Hello everyone, welcome back to my channel. The next topic in continuation of the chapter Immunoassays is Immunofluorescent assay. These are highly specific because they rely on binding of antibodies to specific antigens. The principle of the immunofluorescent assay involves using antibodies labeled with fluorescent dyes to detect specific antigens, proteins, or other molecules within a biological sample. The fluorescent signal emitted by the bound antibodies provide a visual or quantitative readout. It is a powerful technique widely used in immunology, cell biology, microbiology, and medical diagnostics. Immunofluorescent assay can be used for various applications, including the detection of infectious agents, example virus and bacteria, monitoring protein localization in cell, and diagnosing autoimmune diseases and certain cancers. Different fluorophores or fluorescent dyes can be used, each emitting fluorescence at a unique wavelength, allowing for multiplexing and the detection of multiple antigens simultaneously. Immunofluorescent assays are valuable tools in both research and clinical laboratories for their sensitivity and specificity in detecting and quantifying target molecules within complex biological samples. Now let us discuss steps for immunofluorescent assays. First is antigen-antibody interaction. The process begins with the interaction between specific antigens and antibodies. Antibodies are proteins produced by the immune system in response to the presence of foreign substances called antigens in the body. In an immunofluorescent assay, a known antibody that specifically binds to the target antigen of interest is used. Sample preparation. A biological sample containing the antigens of interest is collected and prepared. This sample can be from various sources such as cells, tissues, bloods, or other body fluids. Incubation. The prepared sample is incubated with the fluorescently labeled antibodies. These antibodies, known as fluorescently conjugated antibodies or fluorescent probes, are designed to bind specifically to the target antigen if it is present in the sample. The antibodies are labeled with a fluorescent dye, which emits fluorescence when exposed to specific wavelength of light. Washing. After incubation, the sample is typically washed to remove any unbound or non-specifically bound antibodies. This step helps reduce background fluorescence, ensuring that the fluorescence signals detected is specific to the antigen-antibody interaction. Microscopy or detection. When the fluorescently labeled antibodies have bound to the target antigen, the fluorescent dye is subjected to short wavelength, high energy light, which is absorbed and emitted as light of a different wavelength. The emitted fluorescence has a lower energy than the absorbed light, so the wavelength of the emitted light is longer than that of the excitation light. The fluorescence can be visualized using a fluorescence microscope or a fluorescence detector. Image capture and analysis. In microscopy-based immunofluorescent assays, images of the samples are captured and the fluorescence signals are analyzed. The presence and localization of the target antigens within the sample can be determined based on the fluorescence patterns observed. Quantification. In quantitative immunofluorescent assays, the intensity of fluorescence emitted by the labeled antibodies is measured and can be correlated with the concentration of the target antigen in the sample. This allows for quantification of antigen levels. Types of immunofluorescent assay. They can be of two types, first being primary or direct immunofluorescence and secondary or indirect immunofluorescence. Now let us discuss one by one. Primary or direct immunofluorescence. It uses a single antibody that is chemically linked to a fluorophore. As you can see here in the diagram, this antibody is linked to fluorescein. The antibody recognizes the target molecules and binds to it, and the fluorophore it carries can be detected via microscopy. This technique has several advantages over the secondary protocol because of the direct conjugation of the antibody to the fluorophore. This reduces the number of steps in the staining procedure, making the process faster, and can reduce background signal by avoiding some issues with antibody cross-reactivity or non-specificity. Limitation. However, since the number of fluorescent molecules that can be bound to the primary antibody is limited, direct immunofluorescence is less sensitive than indirect immunofluorescence. Secondary or indirect immunofluorescence. It uses two antibodies, the unlabeled first primary antibody, as you can see here in the diagram, the blue antibody, which specifically binds the target molecule, and the secondary antibody, which is shown here in the diagram in green color, which carries the fluorophore, here fluorescein, which recognizes the primary antibodies and binds to it. Multiple secondary antibodies can bind a single primary antibody. This provides signal amplification by increasing the number of fluorophore molecules per antigens. This protocol is more complex and time-consuming than the primary protocol because it allows more flexibility because a variety of different secondary antibodies and detection techniques can be used for a given primary antibody. Now let us discuss applications of immunofluorescent assays. Antibody detection. It can detect specific antibodies in serum or plasma, which is important for assessing immunity to infectious agents or vaccines responses. Infectious disease diagnosis. They are used to detect the presence of infectious agents such as viruses, example HIV, hepatitis, bacteria, example Lyme disease or syphilis, and parasites like malaria in patient samples. They are particularly valuable for diagnosing diseases with low pathogen loads. Autoimmune disease diagnosis. They are used to detect autoantibodies in patient sera, aiding in the diagnosis of autoimmune diseases like systemic lupus, erythematosus, rheumatoid arthritis, and myasthenia gravis. Cell biology. It is used to study the localization and expression of specific proteins within cells. They help researchers understand cell structure, function, and intracellular signaling pathways. Cancer research. This technique is used to detect specific cancer biomarkers in tissues or blood samples. This aids in cancer diagnosis, prognosis, and the development of targeted therapies. Microbial identification. They are used to identify and classify microorganisms, including bacteria, fungi, and parasites. They are employed in clinical microbiology laboratories for rapid pathogen detection. Virology. They are used to detect and study viruses, including their replication cycles and host interactions. They are vital for diagnosing viral infections. Immunophenotyping. Immunofluorescent assays help immunologists identify and characterize immune cells based on the cell surface markers, enabling the study of immune responses in health and disease. Vaccine development. Immunofluorescent assays are used to evaluate the immune response to vaccines by detecting antigen-specific antibodies in vaccinated individuals. Research and drug discovery. They are widely used in basic and translational research to investigate protein-protein interactions, cellular pathways, and the effects of drugs or experimental treatments on cellular components. Flow cytometry. Fluorescence-based flow cytometry combines immunofluorescence with high-throughput analysis to characterize and sort individual cells or particles based on their antigenic profiles. Multiplexing. Immunofluorescent assays can be multiplexed, allowing simultaneous detection of multiple antigens in a simple single sample. This is valuable for comprehensive disease profiling and reducing sample requirements. Quality control. They play a role in quality control processes such as ensuring the purity and specificity of monoclonal antibodies and other biological reagents. In the next lecture video, we'll discuss ELISA, that is enzyme-linked immunosorbent assay. Thank you for watching.